And all functioning. Excellent. So I've changed the title a little bit from the one which was advertised and moved away a little bit from LRRK2. I'll talk a little bit about LRRK2. That's the gene involved in Parkinson's that I've been working on for well over a decade now. So I'll spend a bit of the first part of the talk talking about that. But really I'm moving a little bit more to focusing on how we identify and characterize new potential genetic loci for Parkinson's. And finding out which ones of them we can target with drugs. So really it's looking to the future. So there'll be a little bit of a retrospective bit, but I'll also be looking to new areas of research towards the second half of the talk. And obviously we're talking about Parkinsonism, we're talking about Parkinson's disease. So it's important that we have an understanding of Parkinson's itself. And we've known about Parkinson's for many, many years now. In fact, over 200 years, 201 years ago, James Parkinson first wrote his essay on the shaking palsy, which was the first description of what we now recognize as Parkinson's disease. And for any of you who are interested in Parkinson's, I really recommend going back and reading his original description, because actually a lot of the description, the clinical description and the clinical observations that James Parkinson made back in 1817 still hold true today. If you talk to a neurologist or a GP now about the cardinal symptoms of Parkinson's, actually pretty much they're the same as the ones that James Parkinson described all those years ago. Of course, he himself didn't name it Parkinson's disease. That was a combination of a pathologist up in Edinburgh during the 1860s, and most importantly, a physician, a neurologist by the name of Jean-Martin Charcot over in Paris during the 1870s. And this is one of the first photos that we have of an individual with Parkinson's disease. This is a gentleman called Pierre, who was an individual with Parkinson's, and it was described as Parkinson's in the clinical notes for this, in Charcot's clinic at the Hôpital Salpêtrière in Paris. And Charcot had a huge impact on neurology across the board. It has, I think, 3 or 4 neurological diseases named after him. But it was really Charcot who brought together our current understanding of what Parkinson's is. And actually, the best way to illustrate this is to look at someone with Parkinson's. This is a video from around 1907, 1908, from a hospital just outside of Brussels in Belgium. And this individual here is in the sort of mid to late stage of Parkinson's disease. And he displays many of the symptoms that most people would recognize as Parkinson's. So you can see that he's got a slowed movement. You can see that he seems very rigid. So if you look at his arms barely move at all. There's almost no arm swing. You can see it takes him actually a long time to turn around. And so the control of movement and of motor skills is one of the areas which is particularly impacted in Parkinson's. Although obviously this is quite an old film, if you look at his hand as he comes back towards the screen, you'll be able to pick out that actually in his right hand there is a slight tremor as well. So there's a sort of continual shaking. And this rigidity, the slowed movement or bradykinesia, and the tremor shown, you can just about pick it out in his right hand there. These are the canonical movement symptoms for Parkinson's disease. And since this time, since the turn of the 20th century, actually we've learned a lot about Parkinson's. We know that if we were able to look inside the brain of Pierre, we know that if you looked in the midbrain, located as you would expect in the middle of the brain, and if you looked at a region called the substantia nigra, so called because of the neuromelanin-containing cells which are contained within this region, which give the area a very characteristic dark color, we know that a huge number of those cells are lost in Parkinson's. And actually, at autopsy, at a neuropathological examination, you don't need a microscope in order to see the damage which occurs within the brain in Parkinson's. We also know that if you look inside the remaining cells in that region of the brain, and actually elsewhere as well, that there are proteinaceous inclusions named Lewy bodies after Frederick Lewy, the neuropathologist who first described them. And really these are a key part of the pathological presentation of Parkinson's. Basically, if you don't have Lewy bodies and you don't have nigral cell loss, the chances are you do not have Parkinson's disease. So from a pathological perspective, these are key to our understanding of the disease. And from the work of Albert Carlson during the 1950s and 1960s, we also know that the key neurochemical deficit that's associated with this cell loss is the loss of this chemical, which is dopamine. And dopamine, and Carlson's work, subsequently recognized by the Nobel Prize, was to define dopamine as one of the group of chemicals that can be used by the brain as a neurotransmitter. And essentially, what happens in Parkinson's is you lose this. You lose dopaminergic neurons, which originate in the substantia nigra, and their connections to other regions of the brain. They are unable to signal with dopamine because of the loss of nerve terminals. And the substantia nigra and the area in the midbrain that it is deeply connected to is very important for initiating and controlling movement. So if you lose those cells and you lose dopamine signaling from those cells, you lose control of movement. And really, that is where the symptoms in the video that I just showed you, that's where they originate from. We've understood this in terms of the pathology since the 1920s, in terms of the neurochemistry since the 1950s. And really, this is where our current treatments for Parkinson's come from. And this is carbidopa-levodopa, which is one of the key treatments for Parkinson's. It replaces dopamine within the substantia nigra. And this is one of the only ways in which really we can manage the symptoms of Parkinson's. Now, one of the challenges with Parkinson's, and this is true across neurodegenerative disorders of old age across the board, really, is that they're becoming increasingly large challenges to health services. This is a paper that was published actually a couple of weeks ago in the Lancet Neurology, which is a systematic analysis of the global burden of Parkinson's disease. And no matter where you look in the world, the burden of Parkinson's disease is increasing. Partly this is due to populations getting older. As the world population gets older, we have more old people. Old age is a key risk factor for Parkinson's. But there is actually more than that going on. We actually think that there is an increase in the incidence of Parkinson's going on as well over time. And interestingly enough, this appears to be the reverse for Alzheimer's disease. So the incidence corrected for population for Alzheimer's disease is decreasing over time. For Parkinson's, it's increasing. Obviously, for Alzheimer's, the number of people with the disease is increasing because more people are getting old. But actually, the corrected incidence is decreasing. We have no idea why either of those are occurring. The thing that really strikes me, in being in a school of pharmacy, I think about pharmacological treatments for disease. The thing that really strikes me is that if you go to the National Institute for Clinical Health and Care Excellence website, which is how the NHS decides what drugs you give people, and you look at the first-line treatment for Parkinson's, that frontline treatment is levodopa. And that has stayed the same since the 1960s. So the drugs that we are using to treat the symptoms of Parkinson's are exactly the same as the drugs that were used during the late 60s, 70s, 80s, 90s, and so on and so forth. We have really struggled to generate new drugs to treat Parkinson's. And it would be very interesting to get an idea as to what Arvid Carlsson thought of this. This is Arvid Carlsson and his Nobel certificate. Sadly, Arvid Carlsson passed away earlier this year, but I think he must have been at least surprised and probably quite disappointed at the fact that a drug that really his initial discoveries drove into clinical practice was still the frontline drug 6 decades later. And this really is where the big challenge is in Parkinson's. So we understand the pathology, we understand the clinical symptoms very, very well. We understand the neurochemistry. We have good drugs to treat the symptoms of Parkinson's, that tremor, slowed movement, and so on. Where we have really struggled is understanding why those cells die, and that's critical. Because if we don't understand why those cells die, we do not have the understanding which allows us to develop interventions which will slow down that cell death and prevent those cells dying. Parkinson's is a progressive disease. The longer you have it, the worse it gets. And so without having a way to slow down or prevent those cells dying in the first place, actually we cannot stop the progression of this disorder. And this is really the challenge that faces the Parkinson's community at the moment. As I say, for many, many years, we struggled to get an idea as to what was causing those cells to die in the substantia nigra. And that really changed about 20 years ago. And the term paradigm shifts and paradigm changes is bandied around science quite a lot. But in terms of Parkinson's, actually you can look back 20 years or just over 20 years now to this paper in Science from Bob Nussbaum's group, studying a family from, they initially looked at in America, but then traced it back to Greece and Italy. And this was the first description of a genetic cause of Parkinson's disease. They described a point mutation, an A53T point mutation in the SNCA gene, which codes for alpha-synuclein. And immediately subsequent to this paper, we were able to identify, and I say we, the Parkinson's community, were able to identify this protein, alpha-synuclein, as a major constituent of Lewy bodies. So this proteinaceous inclusion found within the brains of individuals with Parkinson's disease. And it's been demonstrated since then that mutations in alpha-synuclein increase the aggregation rate of this protein, cause it to be deposited, cause it to become more toxic, and starts causing cell death in the substantia nigra. We're not quite at the point where we're able to reverse that, but at least we're beginning to gain an insight into the underlying mechanisms of disease. And since 1997, actually, we've been very, very successful at identifying Mendelian forms of disease. This is a sort of running list of loci that most people would agree on. Actually, the ones with asterisks are ones which people might not or wholeheartedly agree with. But we're now beginning to generate actually a quite extensive list of Mendelian forms of Parkinson caused by mutations in a range of different genes. And it's one of these that I'll be talking about for the next 10-15 minutes or so. And this is LRRK2, leucine-rich repeat kinase 2. It's a gene located on chromosome 12. And really came to the attention of the Parkinson's research community with two papers in Neuron back in 2004. One from Germany and from the Mayo Clinic in Florida, and the other from the NIH and from the Institute of Neurology in London. Both of these groups identified mutations, coding mutations, causing an autosomal dominant form of Parkinson's disease. And actually, it's one where we have quite a striking public face. And this is quite rarely the case for inherited neurodegenerative disorders. This is Sergey Brin, one of the co-founders of Google. And Sergey Brin has one of those mutations. His mum developed Parkinson's about 12 years ago now. And he's from an Ashkenazi Jewish background for a variety of reasons. The Ashkenazi population has an increased rate of mutations in this particular gene. He had his LRRK2 gene sequenced, and he has a mutation which strongly predisposes him to developing Parkinson's disease. Now, of course, Sergey Brin is one of the wealthiest people in North America, and so he has donated millions of dollars, mainly through the Michael J. Fox Foundation, to study Parkinson's disease, and in particular LRRK2, to try and understand the mechanisms and develop drugs. And some of the research that I'll be talking about over the next few slides is funded by the Fox Foundation. So when you're searching Google, inadvertently, you're slightly funding my research, maybe. I'm actually not funded by the Fox Foundation at the moment, but it kind of works. Interestingly enough, one of the reasons he had his gene sequenced is because his wife at the time, Anne Wojcicki, was a geneticist and subsequently founded 23andMe, the personalized genomics company. And I'll return to 23andMe. If any of you are interested in finding out if you have the same mutation as Sergey Brin, actually you can find out if you send your DNA off to 23andMe. Whether you would want to or not is a very, very different question. So I've been working on LRRK2 since 2005 when I moved to the NIH to work with Mark Cookson in the Laboratory of Neurogenetics. And LRRK2 is a beast of a gene. It's a beast of a gene and it's a beast of a protein. It's a 51 exon gene. It codes for a protein of somewhere around 280 kilodaltons. And it has multiple domains. So it has a number of protein-protein interaction domains indicated here, ankyrin repeats, leucine-rich repeats, and WD40 domain, and an enzymatic core made up of a GTPase domain, or ROC domain, and a kinase domain. And Sergey Brin's mutation is the G2019S mutation shown here. Incidentally, we're now studying worms with the G2019S mutation through Ava Kavai's group here at Reading. But actually, this sort of linear representation of LRRK2 doesn't begin to touch on exactly how complicated LRRK2 is to study. This is our best guess of a model for LRRK2, the LRRK2 holoprotein complex. We think it is functionally active as a dimer. This is a model rather than a crystal structure, by the way. We do not have a crystal structure for the full protein. So you're looking at a complex at a minimum of half a megadalton. And based on a lot of work that's gone into identifying protein interactors, actually we think it's probably a lot bigger than that. At the very least, you've got 2 copies of 1433 interacting there, and probably many other proteins, including substrates and so on. So you're probably beginning to edge up towards a megadalton in size in terms of really understanding the protein complex which is formed from this. Going back many years ago now, going back to 2005, actually we in Mark's group at the NIH, but also a number of groups around the world at the same time, started to try and understand how such a complex protein with multiple enzymatic activities, multiple protein-protein interaction domains could be linked to Parkinson's disease and how it functioned as a cell signaling hub within the cell. There was a sort of working hypothesis which had actually come from a very similar molecule called LRRK2, has a very similar domain organization, but is not involved in Parkinson's. And the idea was that this is operating very similar to the way in which small GTPases operate within the cell. That you have a GTPase domain, a ROCK domain, that basically acts as an intramolecular switch, cycling between GTP and GDP and switching the kinase domain on and off, basically by analogous to what you see with RAS and RAF, which of course has been studied for many, many years because of the role of RAS signaling in cancer. So this was our working hypothesis, and Mark very kindly said, right, we've got a big protein, we've got two postdocs, you take the GTPase domain, and the other postdoc, Lisa Grigio, who's a long-time collaborator and colleague and friend, started working on the kinase domain. And so in a series of papers, really my job was to understand how the GTPase domain of LRRK2 functioned and how it linked into Parkinson's disease. And a number of the initial mutations which were described in LRRK2 actually localized to the GTPase domain. There are 3 or 4 mutations of one residue, an R1441 residue, plus a number of others that fall within the GTPase domain. And so in a series of experiments, moving from simple enzymatic biology through structural biology and then to biophysical characterization, we were able to show that LRRK2 was able to bind GTP and that the mutations in LRRK2 do not alter that binding, so it's not an intrinsic loss of ability to bind GTP. We were able to show using an in vitro GTPase kinase assay that one of the consequences of having a mutation in the GTPase domain of LRRK2 was that this GTPase activity was reduced. And so basically you're reducing the ability of LRRK2 to hydrolyze GTP, converting it to GDP. That would result, we think, in the LRRK2 complex being more active over time. We were able then to show that the R1441 residue actually sits at an interface in a dimeric structure for the ROCK domain, the GTPase domain of LRRK2. So it doesn't sit in the active site. It's not directly altering the GTPase of LRRK2. What we think it's doing is altering the function of the complex. We were also able to show that if you mess around with GTP binding using targeted directed mutations, again by analogy from the RAS field, that you're able to modulate the kinase activity of LRRK2. And that's actually pretty important because that then begins to build into a wider model of how LRRK2 is functioning, with the GTPase domain interacting with and regulating in some way, and even now, 12 years down the road, we're still not sure exactly how that regulation occurs, but there is a direct link between them. And finally, using a combination of differential scanning fluorimetry and secular dichrosum spectropolarimetry, we were able to actually directly look at the stability of the ROCK domain of LRRK2 and what mutations in that did. And what we found was that the mutation which reduces the GTPase activity of LRRK2 reduces the thermostability of that complex. And so we've moved from the clinical observation of this mutation causes Parkinson's through to the molecular defect actually resulting in the dysfunction of that protein. This was actually published back in 2009, 2010, I think, so this is a while ago now. And one of the things that I'm always interested in following up a little bit more is how can we reverse that stability? Of course, the challenge with that is finding small molecules to alter the stability of a protein that work in cells and in vivo is incredibly challenging. The RAS field have been looking at this for about 3 decades, and they've yet to develop a drug which has gone into clinical trials. That tells you exactly how challenging a target the GTPase domain of LRRK2 is. But actually, as we begin to develop the tools to do that, I think this may be an attractive clinical target in Parkinson's. So over the last decade or so, and Claudia Manzoni, who's in the audience here, is responsible for much of this work, we've moved from looking at the biochemistry and biophysics of LRRK2 and the impact of mutations upon that, to trying to understand the underlying cell biology of LRRK2. And what I've shown here is a sort of a cartoon of what a neuron sort of looks like. You've got dendrites up here, you've got the cell body there, the axon shooting along here, and then synaptic terminals over here. The red arrows represent all of the bits of cell biology or neuronal cell biology that LRRK2 has been implicated in. So it's been implicated, and actually I've missed off a few here, it's also implicated in mitochondrial biology as well. It's implicated in the cycling of endosomes. It's been implicated in trafficking, retrograde trafficking actually along the axon, in synaptic vesicle cycling, and in synaptic vesicle release. And really, there's no consensus yet in the field, despite well over a decade of work now, to understanding which one of those is important and which one of those, more importantly, is linked to Parkinson's. Really, what we focused on for about 10 years, actually indirectly funded by Sergey Brin through the Fox Foundation, is trying to understand one aspect of all of that. And in particular, looking at the regulation of the induction of macroautophagy, one of the key pathways that the cell uses to get rid of waste. And for many years, there's been strong links between macroautophagy and waste disposal systems within neurons in the human brain, and the etiology of Parkinson's. The idea being that if your waste disposal mechanisms aren't working properly, the odds are that you'll start accumulating misfolded proteins. Those misfolded proteins will aggregate. They'll eventually form lumps which are big enough to kill cells, and you develop neurodegeneration. And this is well documented in the literature. Not completely understood, but very well documented. And so Claudia, working in the lab, and actually I've cited the scientific reports paper from last year here, but actually this we've been working on this and have published about 4 or 5 papers looking at different aspects of the linkage of LRRK2 to macroautophagy. And really we've taken both a genetic approach and a chemical approach to understanding this. This is a simplified cartoon of the induction of macroautophagy. You get the formation of a phagophore or a megazome. Basically that goes around the cell a bit like a Pac-Man, eating up rubbish and taking it to the lysosomes for eventual disposal. It's under the control of a number of signaling pathways. The ones which are best characterized are the mTOR, URK1 axis, and the Becklin PI3 kinase pathway. And Claudia, in a series of papers, has demonstrated very nicely that LRRK2 is involved in the regulation of macroautophagy. If you use inhibitors which have been developed to switch off the kinase activity of LRRK2, this is using LRRK2/1, one of the first of these inhibitors that was reported, but we've subsequently looked at this using a variety of different inhibitors, that you can induce autophagy using a variety of different markers to measure that. Here we're using the lipidation of LC31 to LC32, but we've actually looked at this in a range of different ways. What she's also demonstrated is that this is independent of the mTOR URK1 pathway. So this is one of the key canonical pathways regulating the induction of autophagy. The data from our group, and actually this has been replicated by a number of other groups now, suggests very strongly that LRRK2 is acting to regulate the Becklin PI3 kinase pathway. Whether this is a direct regulation, i.e. LRRK2 is phosphorylating one of the proteins within this complex and directly regulating it, or whether it's from slightly further afield, and in particular through the regulation of endosomal trafficking to get the complex in the right place to initiate autophagy, actually that's still not clear. And if I were a betting man, which I'm not, then I would say probably the latter. My guess would be that what's happening is that LRRK2 is involved in getting these components to the right place in the cell to initiate macroautophagy, and that is what is really driving all of this. Mentioning inhibitors of LRRK2, of course, allows us to think a little bit more about the translational aspects of LRRK2. And actually, LRRK2 is a very, very good example of how genetic information can lead all the way through to clinical trials. This is a sort of compendium of inhibitors that have been announced and released into the public domain by pharmaceutical companies. So Merck have their PET inhibitor, GSK have released one which is absolutely useless for treating Parkinson's because it doesn't get into the brain, which is why they released it. They had no interest in developing it as an actual therapy. Pfizer have released data on a compound, and Genentech published a while ago now a series of compounds, all of which have been shown to actually target LRRK2 pretty in a pretty specific manner. I'm going to trip over that for a while. I've got 30 minutes, so I've got plenty of time to do that. So these target LRRK2. And most of them are able to get into the brain. So now actually we're in a position to test whether or not switching off LRRK2 has a clinical benefit. We know there are mutations in the kinase domain, the mutation that Sergey Brin has. We know that it increases kinase activity. So a simple test of that hypothesis is to give people with LRRK2 mutations that drug and see whether it results in a slowing of the pathogenesis. And actually, we're beginning to get to that point. Genentech, for a variety of reasons, have run away from LRRK2 research and neurodegenerative research more generally since being bought by Roche. But they farmed out their LRRK2 inhibitor program to a startup called Denali Therapeutics, incidentally run by a number of former Genentech employees. And they have actually taken these drugs into clinical trials. So the GNE 7915, which we've used a little bit as a chemical tool in cell culture to understand LRRK2 function, is now going into phase 1 trials. Actually, the first phase 1 trials, which are summarized here, have completed, and it appears to be safe. They're now running a second phase 1 trial. The FDA, for a variety of reasons, capped the concentration that they could use. So we're beginning to move along that path to really testing whether or not switching off LRRK2 is going to be beneficial in humans. And it's worth pausing there to think about exactly how long that's taken. So back in 2004, the mutations were first identified. Pretty much the next year, we realized the kinase activity was pretty important to this, and G2119S was boosting kinase activity. I was involved a little bit in that, sort of providing a bit of help in the lab to Elisa in the laboratory. I've talked about the GTPase function. We learned about that 2006, 2007, 2008, 2009. The inhibitors, the specific inhibitors, started being released in around 2011. Drug companies started talking about these drugs in 2012, 2013. There was a long period where very, very detailed preclinical tests were being carried out, including non-human primate tests, partly funded by Genentech and then funded by the Michael J. Fox Foundation, which allowed the initiation of the phase 1 trials. And that's, we're now up to 2018. So it's taken 14 years with a gene that we understood pretty well because it had a very clear function, it had an enzymatic function, and one where the evidence was overwhelming. The number of people affected by mutations in LRRK2 is actually pretty high. In the UK, you're looking at somewhere between maybe 2,000 or 3,000 people walking around with Parkinson's because they have a mutation in this gene. But it highlights how long it takes, even if you have all of the resources of a pharmaceutical company and the not insignificant resources of the Michael J. Fox Foundation, pushing and really accelerating research. And this is what I'm going to move on to for the second part of my talk. So we're now at a position where actually we have a number of therapies which are in that clinical trials pipeline. They're either in phase 1 or phase 2 trials. And obviously LRRK2 I've talked about, the inhibitors for LRRK2 are in phase 1 trials at the moment. This is a crystal structure for glucose cerebrosidase, or GBA, another key genetic risk factor for Parkinson's. And there are a number of trials ongoing, which I think are now in phase 2 tests, to see whether boosting GBA activity, GBA is involved in lysosomal biology, whether that has an impact in terms of clinical trajectory, the rate of decline. And this is an NMR structure for alpha-synuclein. And there are now antibodies in phase 3 trials targeting and trying to remove alpha-synuclein from the brains of individuals who have Parkinson's. So we actually have a bit of a pipeline building up here. But if we can learn anything from other neurodegenerative disorders, it's that all of these are going to fail. If you look at the Alzheimer's trials and the process that they've been through over the last 10 to 15 years now, they've had multiple major phase 3 trials fail. And actually the message from that, many of those have been based upon the underlying genetics, is that our expectation is that these will fail and that we will have to either go into further iterations of these trials, targeting slightly different populations, different activities, using different compounds, different approaches, or that we need other candidate targets. And really this is at the heart of a lot of the research that we're doing at the moment in the lab and by collaboration with a number of groups around the world. And where that comes from is really a much greater global understanding of genetic complexity. If you look at Parkinson's disease, probably somewhere around 1 in 20 or 1 in 10, depending upon the population, who develop Parkinson's have some kind of very strong genetic factor contributing to them developing Parkinson's. For the vast majority of people, that is not the case. They do not have an inherited coding mutation or major genetic rearrangement which is causing them to develop Parkinson's. And of course, we're now in a position where we have actually a global map of the human genome. This comes out of the initial sequencing of the human genome and a huge amount of work that's gone on since then to generate a broader understanding of that genetic complexity of complex diseases. And really, I see that analogous in a way to the way in which you can look at a map. So this is a map of a location somewhere in the world. And what we have at the moment is something a bit like this. We have a sort of line diagram of what the genetic complexity contributing to Parkinson's looks like. But actually moving from that to a deep understanding of what that genetic complexity really means is very, very challenging indeed. And really this is what my group and many others are working on at the moment. And this goes back to your chromosomes, really understanding the underlying biology of complex diseases. Clearly, this is something that goes well beyond Parkinson's disease. And there's an excellent review, actually from a few years ago now, almost 10 years ago now, talking about finding the missing heritability of complex diseases. So for Parkinson's, actually, that 5 to 10% of Mendelian disease is really only the very tip of the iceberg. There's a huge amount of other genetic variation which is probably combinatorial, so you need multiple variants to actually go on to develop disease, much of which actually won't cause disease, but will only slightly increase your risk of developing a disorder. And finding that and understanding it has been really the work of the last 10, 15 years since the sequencing of the human genome. Very, very simply, you can take a genome-wide association approach to this, and this is a sort of genome-wide 101. You take a load of people who have your phenotype, could be Parkinson's disease, could be height, it could be intelligence, although I'd query some of the approaches used to do that. You extract their DNA, you take a control population, extract their DNA, and analyze it. Previously, this has been by microarray analysis. Now, increasingly, it's either by exome sequencing or by whole genome sequencing. And you look to see which bits of the genome are more represented in the cases versus the controls, those with your phenotype against those without. And you end up with a Manhattan plot, which basically highlights bits of the genome which are linked to disease. You can do this for any phenotype, basically. So this is a paper from a couple of years ago now, which is actually from 23andMe, looking at self-reporting of being a morning person. One of the questions you're asked with 23andMe is, are you a morning person? So they did a genome-wide association study of this and actually found a large number of loci that were significantly associated with this after correcting for multiple testing. You could also do it for wanting a cup of coffee. So this is a study which actually involves some of our collaborators at the NIH. Again, looking at these massive genome-wide data sets associated with, and looking for loci associated with habitual coffee consumption. And actually, I'll return to the morning person one in a few slides' time. So really what we're trying to do now is move beyond Mendelian genetics and these strong risk factors that cause Parkinson's disease to understanding more common variation that many of us in this room will have. These are common variants that will be widely represented in the human population. And for a number of years now, I've been involved with the International Parkinson's Disease Consortium, which is a global consortium looking to bring together the numbers of patients and controls that allow you to really understand that complex genetic variability. This is actually at Decode Genetics over in Iceland, in Reykjavik, at our meeting earlier this year. And what comes out of this are the pinpointing of associated loci across the genome linked to risk for Parkinson's disease. And over the course of a number of years, this is from 2014, this is actually an earlier iteration of the genome-wide association study. What you get is you get bits of the genome which we think are linked to increased risk for Parkinson's. A very small amount of increased risk. I think at the time, Mike Niles, who's a computer biologist based over at the NIH, figured out that if you combined all of these risk variants and you were unlucky enough to have all of them, actually your increased lifetime risk would be something around the order of 1.5% greater risk than the general population. So clinically, actually pretty meaningless in terms of identifying people who will go on to develop Parkinson's. If you have a mutation in GBA, you've got a 10 times greater risk than the general population. So much, much, much stronger genetic risk. However, what this is doing is it's pinpointing pathways. And so this is the Manhattan plot that came out of that, indicating the bits of the genome that light up in red, which reach genome-wide significance. And fascinatingly, a lot of the genes that came out of this, or the ones which we had an idea of, were genes that we already knew about. Alpha-synuclein over on chromosome 4 was one of those big peaks, and actually the most significant peak after correcting for multiple testing across the genome. LRRK2 is there as well, so we now think that LRRK2 is involved not just in Mendelian disease, but also there's something about the biology of LRRK2 that links to the risk of developing Parkinson's, even if you don't have a mutation in that gene. Actually, that's pretty important for drug discovery, because that gives the pharmaceutical companies hope that if you develop a drug that can treat Mendelian forms of disease with LRRK2 mutations, actually that might be relevant for the rest of the Parkinson's population. I mentioned that 23andMe have kept an interest in all of this, and Anne Wojcicki is Sergey Brin's former wife. This is a paper that came out not long after that genome-wide association study, using their self-reporting data set. So for the IPDGC approach, all of those have been seen by a doctor, and the neurologist was reporting their data. Here, you tick a box to say, I've got Parkinson's disease. Fascinatingly, this data matched almost exactly onto the very carefully calibrated and very carefully controlled data set from the neurologists, which I think the neurologists are now looking over their shoulders and wondering what happens when we develop machine learning a little bit further. Fascinatingly, again, actually, I was talking about the iterative approach that you can go through these. So this is a bioRxiv version of the latest iteration of the meta-analyses for Parkinson's disease. This is Meta 5, so it's the fifth meta-analysis that's been carried out, and this is actually beginning to really drill down into all of that genomic data. We now have over 40 loci across the human genome, which we think are significantly involved in genetic risk for Parkinson's. Let's put that into perspective. For the Mendelian forms, we have somewhere between 15 and 20, depending upon how you define Parkinson's. We now have an additional 45 to 46 new loci, and that number will increase as time goes by. Importantly, this comes out of looking at almost 40,000 people with Parkinson's, extracting their DNA, analysing it, and over 1.4 million controls. So that gives you an idea as to the scale of this kind of analysis. I said I'd return to the morning person study, so this is also on bioRxiv. I stumbled across this when I was putting together the slides for this. So actually what they've done is they've combined that data from the morning person genome-wide association study and used a very interesting technique called Mendelian randomization, which allows you to basically chop up the genome and carry out in silico analysis of how that genetic risk localizes. And actually they think that the tendency towards being a morning person, self-described, increases your risk of Parkinson's disease. So that shows actually the power of a sort of signposting what to look at in terms of clinical and non-clinical phenotypes. Of course, it doesn't give us any understanding of the underlying molecular mechanisms. And this is tough. Betraying my complete lack of a classical education, you can sort of think of this as either being Herculean, and this is Hercules fighting Cerberus in the underworld, or Sisyphean. This is Sisyphus, who was punished by the Greek gods to push a rock up to the top of a hill, only to watch it tumble down all the time. And in a way, you can think of the efforts to understand these genome-wide loci as being actually like a combination of these, because the vast amount of potential genes to look at, the cellular processes, the details that we need to get out of all of that is phenomenal. This is a massive challenge. This is true for Parkinson's. It's also true for any other complex disease where you have this kind of data set. To give you an insight into what that means in terms of getting that kind of understanding, we've worked for 14 years on LRRK2, and it's taken millions of dollars, hundreds of laboratories working across the world to understand the cell biology of that. We understand LRRK2, we understand GBA, we understand synuclein. That's 3 out of the genes that have come from Mendelian studies. We now have 40-odd other genes, and we have no idea how they link across to Parkinson's. Really, we understand 3 of those genome-wide association sites, and actually, that's synuclein, LRRK2, and GBA, the ones we already knew about. So we now have to replicate the effort that's gone into the studies for LRRK2 and synuclein, or GBA, but at a genome-wide scale. And to give you an idea as to how complex this data can be, this is the LRRK2 locus highlighted from the recent genome-wide association studies. And basically, each of these dots represents a single nucleotide polymorphism. And the ones which reach reach significance for genome-wide association are basically above a line somewhere around here, 10 to the minus 8. So what we have is we have a lot of genetic information saying that there are changes at the LRRK2 locus involved in Parkinson's, but actually we don't understand what those changes do, and it has taken about 5 years even to get the slightest hint as to what's going on there. This is Dania Trabzuni, who's a senior postdoc over at the Institute of Neurology. And she's been working for 5 years trying to understand what those single nucleotide polymorphisms do, initially funded by the Fox Foundation, now by the MRC. And what she's found is that basically no matter what we look at in terms of expression of LRRK2, really there is not a significant alteration in the brain. We're looking across data sets for gene expression in different brain regions from just different genetic backgrounds, using a lot of approaches to try and understand gene expression, and we don't think that it's a crude alteration in gene expression. The best guess is that it's altering some level of splicing. This is actually the splicing out of an exon in the substantia nigra in the brains in the human brain. What we think might be happening is actually there's an alteration in splicing. This is very challenging to look at in LRRK2. You've got 51 exons. Trying to understand that across the whole of the gene is tough. But actually, we have gone on to look at some of these other loci as well. This is Megha Murthy, who was a student from India, working funded by the Newton Fund. And she took one of those other loci. She took a region on chromosome 7 where we didn't know which gene was involved in Parkinson's, and looked at gene expression. And so for some of these loci, we're beginning to get an insight as to what's going on. I'm working with Dania. Megha was based here at Reading, but worked in between here and UCL, and analysing that same spread of risk SNPs across the genome. She was able to identify GPNMB as the gene which is most affected at this locus in terms of gene expression. And we now think that an increased expression of this gene at a very, very subtle increase over your lifetime does actually increase your risk of developing Parkinson's. And again, that gives us the beginnings of a pointer towards understanding the biology of all of this. If you understand which direction a gene is being altered with this risk, actually you can begin to understand what is helping cause the disease. And there are a number of papers that have come out since Megha published her study on this, highlighting GPNMB as being involved in inflammatory response, in particular within astrocytes in the brain of individuals with Parkinson's. And this is now actively being investigated by a number of pharmaceutical companies. So the pharma companies are beginning to look at those loci, trying to understand the biology and trying to find ways to target it. And really this is what my group over the next several years is going to be working to try and understand. So hopefully what I've done is talked a little bit about how we can go from, obviously I've talked a little bit about LRRK2 at the start, but I've talked for the last 10-15 minutes about how we can go from that sort of understanding of the data underpinning genome-wide association to beginning to actually be able to visualize what that looks like. So out of curiosity, does anyone know where this is? And anyone who's seen this talk before is not allowed to answer. Just to give you an idea as to how challenging it can be to visualize all of that. Any ideas? Any guesses? You're getting close. Geographically, not far away. So it's actually the Yosemite Valley. So what you're looking at there is a two-dimensional representation of the grandeur of the Yosemite Valley, one of the most breathtaking places on Earth. One thing I would say, with apologies to Ansel Adams, is that actually you can take pretty good photos of Yosemite Valley no matter what camera and no matter your level of skill. Obviously Ansel Adams was a bit better than my snaps on my phone, but the Yosemite Valley really illustrates for me the difference between understanding the data underpinning something and the realism, the real representation of that in 3 dimensions in the world. And this is the challenge that we have with Parkinson's disease. It's true of all complex diseases where we're taking this genome-wide associated approach. But really, this is what we're trying to do. We're trying to take that data and push it through to a detailed 3-dimensional understanding of how these genes are involved in Parkinson's. With that, I'll finish. Obviously, this has involved a lot of different people around the world and a lot of different funding bodies. I'd like to highlight in particular Claudia Manzoni and Daniel Trabzuni, who worked on the cell biology and the gene expression of LRRK2, but also collaborators across the globe who've worked on this, and obviously mainly funded by Parkinson's UK, the MRC, and the Fox Foundation. I'm more than happy to take any questions at all.